tissues of internal medium group, connective, proper, skeletal, blood, muscle. Now the last type of tissue. Only one tissue forms this group, nervous tissue. Nervous tissue. Who can remind me what components present in nervous tissue? What are the structures that we can find in the nervous tissue? No? What? First of all, neurons. Nervous cells. Neurons. These cells are the main in the nervous system. Why? Because these cells generate nervous impulses, conduct nervous impulses, analyze information, and regulate the functioning of another tissues and organs. Yeah? So neurons are the main component of this tissue. But besides of the neurons, if only neurons present in the tissue, in the organs, nervous tissue forms nervous organs of the nervous system. Brain, spinal cord, peripheral organs, ganglia, nerves, all these organs contain nervous tissue. Neurons. But if only neurons present in the organs, the functioning is possible or impossible? Glial cells, obligatory. This tissue, besides of the neurons, nervous tissue contains obligatory another cells, glial cells. Different types of glial cells present in our nervous system. And according to morphology, according to the function, position in different sides of the nervous system, we can classify the neurons and the glial cells. Yes? No? Morphological. What are the morphological peculiarities of the neurons? Why bipolar? Only bipolar? No. Any neuron. What is a morphological feature of a neuron? This cell has processes. Neurons have processes. Obligatory. How many processes? Three. What? And axon, dendrites, and dendrite cells. No. Number of processes may differ from one to numerous. If a cell, if a neuron has only one process, how we name this neuron? According to classification, morphological classification. Neuron with one process only. Multi? Bi? Uni. Unipolar neuron is a neuron with one process. But what process present obligatory in any neuron in unipolar too? Axon. Axon. If process is single, it means axon present. Axon present in any neurons obligatory. Axon process presented constantly. If one process present, it means axon present. But in human organism, this type of neurons absent. Unipolar neurons absent in our organism. In unvertebrate species, such as mollusk, for example, we can meet unipolar neurons in mollusk. But as me, I am not mollusk. I have no unipolar neurons. You too, be sure. Another type of neurons present in human organism. What type of neurons you know else? Look at this chart. See you. The chart demonstrates different types, different morphological types of neurons. From the bottom to the bottom, да? From the top, oh, sorry, from top to bottom. From the top to bottom. What types of neurons see you? The upper neuron. Uni. Bipolar. Look, there are in our organism, there are the neurons that look like unipolar under light microscope. Some neurons look like unipolar. So it seems that one process begins from the cell body named perikaryon. Perikaryon is the cell body of the neuron. Perikaryon means around the nucleus. Nucleus in Greek, nucleus is karyon. Perikaryon, so cytoplasm around karyon, nucleus. Cell body of neurons is perikaryon. Terminology. Okay? So, it seems, under light microscope, it seems that one process begins. And then this process branches. But it seems, because in reality, when we examine this neuron under electron microscope, we can see that two processes begin from the same region of the perikaryon. Two processes. And the initial portion of these processes is surrounded by the same glial cells. And then each process go in different direction. So one of them is dendrite, another one is axon. So in reality, this neuron is bipolar. In reality, because it has two processes, dendrite, axon. But these two processes, dendrite, axon, begin from the perikaryon at the same site and initially go together and then ramify, separate. Is that clear? These neurons present in our organism, and we name these neurons, demonstrated in the top of this chart, pseudo unipolar. Pseudo means what this pseudo. False. Pseudo is false. Pseudo unipolar neurons. Pseudo unipolar neurons present in peripheral organs of the nervous system, not in the central organs. In ganglia. In what type of ganglia? In sensory ganglia. Sensory ganglia, we will examine today the slides. Sensory ganglia contain pseudo-unipolar neurons. And if we compare the morphology and function of these neurons, we classify neurons according to morphology, according to the number of processes, and we can classify the neurons according to their position in the chain. How we name the chains of neurons? Do you know that all neurons contact? Yeah? Neurons are not scattered in nervous system, are not separated. Normally neurons contact to form the chains of the cells. And these chains of the cells we name as chain of the neurons. What? PNS. PNS is peripheral nervous system. No. see a chain of nervous cells. Together, all cells, all neurons that form this chain correspond to so-called reflex arch. Arch. Reflex arch. Reflex arc. Reflex arch. So chain of neurons forms reflex arch. And any reflex arch may be simple, containing only 2 neurons, 3 or more neurons. These neurons may form reflex arches. But any reflex arch contains some specific types of neurons. The first neuron in any reflex arch, what neuron is the first in the reflex arch? Sensory neuron. Sensory neuron. Afferent, receptive, sensory, synonyms. So the first neuron in the reflex arch receives information from external or internal environment. It's receptive neuron. The second one may be motor, if reflex arch contains 2 cells, but there are interneurons or associative neurons too. If reflex arch has 2 neurons only, one of them is sensory, another one motor, efferent, motor, afferent, efferent, sensitive, motor. But the most of reflex arches contain interneuron, associative neurons between receptive and motor. You must know well reflex arches. You must explain the principle of organization. You must draw the reflex arches. Obligatory. So, and pseudo-unipolar neurons. Pseudo-unipolar neurons are localized usually in the peripheral ganglia, localized at nearest of the spinal cord. Sensory ganglia named spinal ganglia or posterior root ganglia. What is this ganglion? When we say ganglion, it's organ, peripheral organ. It's organ of PNS, peripheral nervous system, organ. When we say ganglion, what does that mean, ganglion? What is this ganglion, this organ? Huh? Situated outside of the central nervous system, it's correct. But what are the components of ganglion? Cell bodies. Yes. If you see neurons, cell bodies, pericardia of neurons, that are grouped in some region, it means you see ganglion. Ganglion is a group of neurons localized in some sites, in some organs. Ganglion is group of neurons. So, you see anatomical structure named posterior. It's a half of the spinal cord. The scheme demonstrates somatic reflex arch. A half of the spinal cord, posterior root, anterior root. I know that you didn't study yet anatomy. So maybe you know that the nerves, peripheral nerves, can come to the spinal cord, and some nerves begin from spinal cord. These nerves are named as roots. Posterior root enter in the spinal cord, anterior root go out from spinal cord. Posterior root, anterior root. When two roots, these two nerves, posterior root, anterior root, fuse, spinal nerve appears. Spinal nerve and two branches. One branch go to the spinal cord, another branch go out from the spinal cord. Is it clear? And now look, in the posterior root, in this anatomical structure named posterior root, there are group of neurons, and this group of neurons corresponds to ganglion. Clear? So, ganglion localized in posterior root is posterior root ganglion or spinal ganglion. And pseudo-unipolar neurons that are sensory neurons are localized in this ganglion. So if I ask you, remind me please, where pseudo-unipolar neurons may be situated in our organism? What will you answer? In spinal ganglion. In spinal ganglion. These ganglia are sensory because neurons, pseudo-unipolar neurons, are the first in the reflex arch. These neurons are receptive, are afferent, receptive. Is it clear? So, the first morphological type of neurons, pseudo-unipolar, are usually sensory and are usually localized in the ganglia situated at nearest of the spinal cord, in posterior root, in spinal ganglia. Clear? Keep in your mind. Next morphological type of neurons presented in this chart, in the middle. In the middle. Bipolar. Real bipolar. Pseudo-unipolar is bipolar, but pseudo-uni, yeah? Here, bipolar. You see that one process begins at left, another at right. So, bipolar, two poles, yeah? One of them, one process is axon, another process is dendrite. But this process begins from different sides of the pericardium. In pseudo-unipolar in the same region, bipolar in opposite sides usually. Is it clear? These bipolar neurons, where these bipolar neurons may be disposed in reflex arches, and in what organs we can meet bipolar neurons? No? Who remember? Bipolar neurons may be sensory too, may be receptive too. The most of sensory neurons are pseudo-unipolar, the most. But some receptive sensory neurons may be bipolar. And we will meet these bipolar sensory neurons in the retina. In the retina of our eye, there are receptive cells, photoreceptive cells, it's neurons, and these photoreceptive cells are bipolar, not pseudo-unipolar, but bipolar. So bipolar neurons may be sensory and situated in the retina. But some of bipolar neurons may be associative too. And associated bipolar neurons we will meet in the retina too, in the same organ in the retina. So retina we will study the eye in 2 weeks, and you will see that retina, it's tunic of eyeball. Retina contains numerous neurons, 3 types, 3 neurons, chain of neurons. The first will be receptive, the second one will be associative. Both are bipolar. Clear? So, pseudo-unipolar neurons, only sensory. Bipolar may be sensory or associative. But the most of associative neurons in the central nervous system are multipolar. So multipolar neurons demonstrated at the bottom of this chart, contain more than two processes, numerous processes, but one of them is obligatory exon, and the rest, two or more, are dendrites. These neurons are multiple. Where these neurons is situated? In the most of regions of our nervous, the most of neurons in our nervous system are motor neurons. Motor neurons may be associative. Motor neurons are obligatory. Multipolar neurons. Multipolar neurons may be associative. Multipolar neurons are usually motor, efferent. Look at this scheme once more. Pseudo-unipolar sensory in the ganglion, associative multipolar in the gray matter of the spinal cord, and motor neuron is multipolar too, situated in gray matter. So, morphological and functional classification of neurons and position, localization of different neurons, different according morphology, different according their function, their position in reflex arch, yeah, you must keep on your mind. And if you obtain the section of the cerebral cortex, for example, we will study next week this slide. Cerebral cortex. What types of neurons present in cerebral cortex? Morphological types and functional types. In the brain, what neurons? Sensory, associative, motor neurons present. Huh? No, sensory in peripheral ganglia. Sensory outside of the central nervous system. Motor for somatic. In spinal cord. In spinal cord, red neuron, see you? It's motor neuron. In spinal cord, in anterior horn of gray matter. In the spinal cord. In the brain, no sensory, no motor. All neurons in our brain are associative, interneurons. So what is the morphological type of neurons in the interneurons? Associative. I explained already. Associative neurons may be bipolar in the retina, for example, but the most of associative are multipolar. So in the brain we will have multipolar associative. If we examine spinal cord, we will examine today this section too. Look, spinal cord has white matter peripherally, situated peripherally, and gray matter in the center, yeah? And in the gray matter there are pericardia, there are neurons. Processes are situated in white matter. Gray matter is a substance containing pericardia of neurons. Cell bodies are situated in gray matter. And look, in the gray matter, in the spinal cord, what neurons? Interneurons, motor neurons. What is morphological type of these neurons? Pseudo-unipolar, bipolar, multipolar? No? Multipolar. My dear students, once more, pseudo-unipolar. Only in sensory ganglia, spinal, for example, yes. Bipolar, may be sensory, may be associated, but where? Peripherally, in eyeball, for example. The rest neurons, all are multiple. Multiple. So, I show you spinal cord. You don't know the anatomy, you don't know history of this organ, but you know the principle of position. In the spinal cord, what type of neurons must be present? Multipolar only. But some of them may be motor, some of them may be associated. In the brain, only multipolar, but only associated. Is it clear? Huh? Shemini? Clear? Good. Besides of the neurons, with processes, axons, dendrites, there are another cells in our nervous tissue, in the organs of the nervous system. Cells of glial. Glial cells. And how we classify the glial cells? We can classify because glial cells differ. That's why we can classify, yeah? Subdivide the cells on the groups. No? What is? Who knows classification of glial cells? Macro. Two groups. Macro. Macro contains oligo, estro, ependymal glial cells. Three types of glial cells form this group. Ependymal, estro, and oligodendrocytes. In what parts of our nervous system we can meet different types of glial cells? For example, ependymal glial cells. Ependymal glial cells. Central nervous system or peripheral nervous system. CNS, VNS, where? CNS. Why? Because ependymal glial cells line the surfaces of the cavities presented in our brain or in the spinal cord. In the spinal cord, there are central canal. Central canal, you see a part of this canal here. Central canal in the spinal cord. It's a cross section of the spinal cord. And see you, central canal. Cavity. In the brain, there are ventricles. First, second, third, fourth ventricles. And the wall, surface of these cavities, ventricle, canal, surfaces are lined by sheet of cells, by a layer of cells, I'll show you this. These cells. Look. Oh, see your cavity. It's central canal of spinal cord. It's gray matter of spinal cord and the layer of cells. No, it's not Schwann cell. It's ependymal glial cells. It's not Schwann. Ependymal glial cells. Cells are prismatic, tall, contact together without any intercellular matrix. Cells are polarized. On the apical pole there are cilia. The basal pole has process that penetrate in the gray matter. So what tissue reminds this layer? Epithelium. This layer of cells reminds epithelium, but it's not epithelial tissue. Absence of basement membrane, and moreover, absence of keratin in cytoskeleton. Do you remember? Epithelial cells contain cytokeratins, proteins in intermediate filaments of cytoskeleton, cytokeratins. These proteins present in epithelial cells. Keratin. Here keratin absent. Acid fibrous, glial fibrous protein present in this cell. Another protein. So it's this tissue, these cells remind epithelium, but are not epithelial cells. Is it epithelial-like layer of ependymal cells. And why these cells, this sheet of cells reminds epithelium? Because function is similar. What is the function of epithelium? Barrier. Epithelium is barrier that realize exchange between different media. Yeah? Here you see cavity filled with fluid, cerebral spinal fluid present or liquid present in the cavity, and the tissue of spinal cord. So between across of these ependymal glial cells, exchange is realized from fluid, from liquid to tissue, from tissue to liquid. Transport, barrier, transport, exchange, functions are similar with the epithelium. That's why morphology of ependymal glial cells is similar to epithelium. But it's not epithelial tissue. These cells belong to nervous tissue. Clear? So, ependymal glial cells present in CNS, especially in the region where cavities present, ventricles or canal of the spinal cord. Astral glial cells. Astral glial cells. Astrocytes. In what part of our nervous system present astrocytes? Astrocytes may be protoplasmatic, fibrous type. Do you remember? So different types of astrocytes are situated in CNS, right? But moreover, not only in CNS, but in the gray matter of CNS. CNS, brain, spinal cord, consists of the white matter and gray matter. Glial cells, astrocytes, are situated in gray matter, in the substance where neurons, cell bodies are situated. Gray matter is the region where cell bodies of neurons. So astrocytes are the structures, are the cells of gray matter of CNS, not white matter, not PNS. Oligodendrocytes. Oligodendrocytes. Where oligodendrocytes may be found? Anywhere. In all organs of the nervous system. CNS, PNS, in ganglia, in peripheral nerves, in white matter, in gray matter. Why? Because oligodendrocytes surround the cell bodies, the pericardia, as well as processes. Look, see you, cells localized along of the processes. And around the pericardium, glial cells, oligodendrocytes, present too. So, imagine, I am a neuron. It's cell body, pericardium, it's processes. All my cell body is surrounded by glial cells, as well as processes. Process is longer. Axon dendrite may be very long, some dozen centimeters. Oligodendrocytes are small. That's why numerous oligodendrocytes surround this process. Clear? And where processes of neurons may be situated? Look. Let's examine once more our skin. Look. Cell body in the ganglion. It's peripheral organ. Peripheral organ. Pericardia present here, present. Around pericardia, around cell bodies, oligodendrocytes present, present. And these oligodendrocytes that surround cell body, look, you see here, neurons, it's a sensory ganglia. What type of neurons present in this sensory ganglia? Pseudo-unipolar, and functionally? Sensory. So you see cell bodies of sensory neurons, and around of each neuron you see the cells. What cells surround? Oligodendrocytes. How we name these oligodendrocytes situated on the surface of pericardium? Satellite cells. Oligodendrocytes around the pericardium are usually named as satellite cells. It's oligodendrocytes, but satellite cells. And now, tell me please. Tell me please. Cell body of this neuron is situated in the ganglia. But this process and this process go along of the nerves, peripheral nerves. And when process is surrounded by glial cells, what structure is formed? By the process and the glial cells around. How we name these structures? Containing axonodendrite with glial cells around. How we name these structures? We name these structures nervous fibers. Nervous fibers. So nervous fiber is a complex. Process and glial cells around. It's nervous fiber. And numerous nervous fibers, bundles of nervous fibers, form the nerves, organs, anatomical organs. Yes? And now look. Process in the peripheral nerve. But around this process, there are glial cells. What type of glial cells? Oligodendrocytes. But we name these oligodendrocytes compared to satellite cells. These oligodendrocytes are Schwann cells or lemocytes. Terminology. Understand? Look, once more. Neuron, processes, and glial cells around. What glial cells are situated at nearest of neuron? Oligodendrocytes. Oligodendrocytes around pericardium may be named as satellite cells, and oligodendrocytes along of processes are usually named Schwann cells, because Schwann described earlier, or lemocytes. Lemocytes. It's oligodendrocytes. Clear? Now tell me please once. Stop. Look at this part of the scheme. Look. Neuron, pericardium, process. Pericardium, process. So, processes present in gray matter and in white matter. So, where we can meet oligodendrocytes in our nervous system? All around of this system. In peripheral, in central, in gray, in white matter. Because we have neurons and processes. Neurons and nervous fibers. All of them are covered by glial cells, oligodendrocytes. Clear? So, ependymal only in CNS, only in cavities. Extra in CNS in gray matter. Oligo in all regions. What about microglial cells? Microglial cells. Microglia. What is this? What is the function